Very pleased to introduce our next speaker, Jean Johnston. She is the data banking manager for CODIS, the Washington, which is a part of the Washington State Patrol Crime Laboratory Division. She's analyzed evidence in hundreds of forensic cases, including key DNA evidence in the Green River murder, and regularly testifies in court as an expert witness. And when we were looking for an expert to present to you all, Jean was mentioned as one of the best. Her master's degree is in forensic chemistry from the University of Pittsburgh. Her initial work was in forensic science at the Houston Police Crime Laboratory. And in 1980, she was hired by the Washington State Police Crime Laboratory as a forensic scientist specializing in biochemistry and DNA analysis. She's a member of the American Academy of Forensic Scientists and a diplomat in the American Board of Criminalities. And we're very pleased that you can be here for us this evening.
In my talk tonight, I'm going to be talking a little bit about what DNA is, just sort of briefly, and then also how we use it in forensic science. I will also touch on a program that I'm currently the program manager for the state of Washington, which is CODIS. CODIS is an exciting tool that we are able to use. It basically kind of marries up the DNA data analysis that we're able to generate out of evidence samples and use computer software technology. We can provide then information to the criminal justice community on who possibly could have been a perpetrator of a crime, even on crimes that they have no known suspect on. Then to end up my talk tonight, I'm going to talk about one of the cases that I've worked on over my years in the crime laboratory, and I personally just find this one particularly satisfying, so I'll be sharing it with all of you. Okay, so let's just get in with what is DNA a little bit. DNA is the acronym for deoxyribonucleic acid. It's that basic molecule of inherited information, and you get half of it from your mother and half of it from your father. There are 23, it's arranged in 23 pairs of chromosomes in your body cells, and this is found in the nucleus of each of your cells. All of your body cells have nuclei except for your red blood cells. So when you hear me talking about typing DNA out of a blood stain, kind of an interesting little tidbit is it's not coming from the red blood cells, but it's coming from the white blood cells that we're looking at. The DNA basically kind of looks like the schematic up here at the top of the slide. It's a double helix, and it's double-stranded. This schematic is just to kind of help you give you a visual of what I was talking about. So this is the cell nucleus that I was referring to. It's kind of the control center of your individual cells, and it has all the information kind of needing to get your body going and working properly. And within that cell nucleus are your 23 pairs of chromosomes. And the chromosomes then are made up of these individual strands of the DNA molecules, all wound up very tightly coiled and protected in chromosomes. And then we're actually targeting down and getting into these individual nucleotides when we're typing the sample forensically. So why use DNA in forensics? It's unique. All individuals will have a different DNA pattern that we can exploit forensically, except for identical twins. And your whole genetic makeup that you have, all your 23 pairs of chromosomes, is called your genome. And 99.9% of all humans, their genome is exactly the same. But forensically, we exploit that 1/10 of 1% and are able to come up with individualizing information except for identical twins. All nucleated cells in the body do have DNA, as I mentioned earlier, and all of those nucleated cells within one person's body would be the exact same type of DNA. So it's the same, consistent, there's lots of it in the body. Because of the double helix structure, DNA is very stable, which is very important to us forensically because we really never know what type of evidence we're going to get in the crime lab. We're not a quality control type lab where you get a routine sample always coming in, like always a blood sample that's in a nice tube and has been kept refrigerated. Many of our samples have been out and exposed to the environmental conditions for days, months, or years. So it's very important that the DNA is very stable. And the most powerful thing over the past decade or so is we are now able to type DNA on very minute samples, just such as small as like a single hair root. And I'll be going into the technology that was developed so that we can do that at the present time. This is just a slide to give you a little bit of example of the wide variety of forensic sources that do have human DNA in them and that we encounter in the crime laboratory. You will hear blood stains being typed and semen stains from, say, a sexual assault case. Those are typical types of DNA generating materials that you might also see on TV, etc. But something you might not think about is like on a bludgeoning case and the suspect is wielding the hammer like Corinne was talking about earlier. We've had some real big successes with looking at the suspect's shirt microscopically. The suspect has no idea that he's carrying around pieces of brain tissue from somebody else, but we're able to look at it microscopically and find very small particles of tissue and able to extract DNA off of something like that. Hair roots, we're able to get a DNA typing profile. Saliva has a large quantity of DNA in it. And then the tooth material, specifically the pulp and then the bone, are samples that we sometimes will get into the crime laboratory. Usually those are going to be associated with a set of unidentified human remains, a skeleton that's perhaps been out at a scene for a period of time, and there's maybe not a lot of identifying material on it, so you just have the bone or the pulp teeth pulp to work with. We can generate a DNA profile off of that and hopefully provide an identification then. Now, besides those traditional sources that I was just talking about on the previous slide, we can also get DNA from just shed skin cells. So that's just a really powerful thing that we're able to do now with the newer technology of DNA typing we're using forensically. And these are some of the different examples of sources. So in the past, if you had a knife that was submitted to the crime laboratory, the forensic scientist's mind is thinking about looking at the knife blade to see if there's some blood on there that maybe came from the victim. But now we're also turning our sights and looking at the knife handle to see if we can determine whose DNA is on the handle of the knife that perhaps was holding it when the victim was stabbed, and we can associate that perhaps with a suspect. Some of these other exhibits on here, same type of thing with the grips or the trigger area of a gun. Oftentimes you won't be able to get a latent fingerprint off the grips because it's got a textured surface there, but we certainly might be able to get a DNA typing profile off of that. This slide over here is tape, and duct tape is a forensic scientist's dream because you can oftentimes either get saliva off of that because the criminals will they'll stick their mouth on it and try to tear a piece of it off. So saliva, like I said, is a really great DNA carrying body fluid. And even if we can't isolate some saliva off of that type of tape, then we might be able to get skin cells where they've taken the tape and ripped it apart like this. And fingernail clippings, I think Corinne also mentioned that that's a regular part of what would be collected during an autopsy. We would be looking at those to see if we can get some DNA transfer of skin cells that might have happened during some type of assault and altercation. so that the skin cells from the suspect are going to be transferred over onto the victim. These other two slides are examples of ligatures. So the clothing item, pantyhose, cord, whatever is used, is going to be pulled very tightly by the suspect up against the victim's neck. And around the cord, it's usually still in place. So you don't usually have the question of, well, whose DNA is on that portion of the ligature. But you will oftentimes be able to tell who the suspect might be by getting the DNA profile off of the ends of it where the suspect has pulled very tightly. And his and it's usually a guy, sorry, but I'll refer to it as a his DNA would be transferred onto the ends of the ligature. So that's another area that we oftentimes target. And this is called cellular or transfer type DNA. There are 5 basic steps to the DNA typing process that we use in the crime laboratory currently. The first step is to extract the DNA from the component or the medium that it's on. As I said, usually we don't get in nice pristine samples into the crime laboratory, so we could get a variety of different kinds of stains, and we never know if it's going to be on something as simple as an article of clothing. It might be on a weapon, rocks, or a gun. We've had whole doors submitted into the crime laboratory, car hoods, et cetera. So we need to get the possible DNA material off of that substrate that it's on. Now the next step is quantitating the DNA that we have present. Just by looking at a particular stain, you cannot really tell how much DNA is in that particular stain, because there's quite a wide variety of the concentration of DNA in various body fluids. So you'd have quite a high level of DNA in something such as a semen stain on a rape case. It doesn't take that much spermatozoa for us to be able to get a DNA typing profile. But if you have a urine stain that's in question, and it's not uncommon for there to be urine left at something like a peeping Tom type incident or something. So occasionally we do get urine samples into the crime laboratory to test. There's not a lot of cellular material in those. So we need a lot of liquid there to be able to get a DNA typing profile out of that type of sample. The third step in this process is to amplify up the DNA, and this is making many copies of the original starting DNA that we had to work with. The fourth step is detecting the DNA using an instrumental process. This is separating out the different DNA fragments that we have present. And the final step is using some computer software to actually interpret, do the data analysis of the information that you received off the instrument and come up with a DNA typing profile. So that first step is going to be taking that small amount of the evidence stain. The DNA is extracted from the cells, and for the type of technology that we're using in the crime laboratory right now, we only need about 1 nanogram of DNA to work with. And it just probably seems like a number to you, just like, well, I don't know what that is. And before I was in the crime lab, I really didn't know what that number was either. It's not something when you go into the grocery store, you're going to go shopping for an anagram of something. So this is sort of a visual for you. A dollar bill weighs about 1 gram, and you would have to cut that dollar bill up into 1 billion pieces, which clearly is something that you can't see or do. And that is, we just need 1 of those. So 1 billion here is 1 and then 9 zeros following it. So we're getting to have a really, really small amount of sample of the extracted DNA that we need. Once we've extracted and done the quantitation step and we're ready to progress to the next stage on our DNA typing process, we do the amplification. And this is the breakthrough that happened back in the mid-'80s. A gentleman named Dr. Kerry Mullis won the actual Nobel Peace Prize for this process called, and it's basically sort of a molecular Xeroxing or molecular copying of the original DNA material. It's done on an instrument called a thermal cycler, and the thermal cycler basically can be programmed to be set at various temperatures over a period of time. So we have it go to controlled temperatures where specific reactions will happen to the starting DNA in there. And we have added some building blocks of DNA components. So the starting DNA can add these building blocks at the different temperatures and then make more of the very same starting DNA that they started with. And this PCR process, or polymerase chain reaction, is actually the powerful tool behind us now as forensic scientists being able to get a DNA typing profile off of something so small as just a single hair root, or just like a little tiny piece of glitter, about that size of blood is all we would really need. PCR is also a tool that's used in a wide range of applications. It's not just used forensically. It's used in the medical field. It was used to help type in the Human Genome Project. They use it in anthropology. They use it to do animal pedigrees, agriculture, etc. So it has a well-based science application. This is a visual of the polymerase chain reaction. And like I said, you'll start out over here with your double-stranded DNA, and it goes through a denaturing cycle where the DNA strands will separate into two, and then they add the building blocks in there, and so those two then become 4 strands of DNA, and then the 4 strands become 8, and etc. And over a period of time, it usually takes about 3 hours for the process for this to work in the crime laboratory. We end up with about 1 billion pieces of DNA of the same starting type of DNA that we started with. So it's through that process that we're able to get into the next stage of the detection. And we have enough DNA to the level now that we can detect it by going through the PCR process. We use an instrument in the crime laboratory called a capillary electrophoresis, and it's basically just using a very small wire. It's a little bit fatter than a hair that the DNA solution is injected, a voltage is applied to it, and the DNA solution goes through this capillary, and the small pieces of DNA go through there very, very fast. And those are read and captured by a laser and a camera mechanism, and then it's recorded into a computer software program. And then as it progresses through the DNA that's in our little tube there, the little bit more heavier type of DNA fragments will come out in the medium levels, and then our very slow ones come out at the end. And for one single sample to come through the capillary electrophoresis unit, it takes about a half hour. I'm going to take you in to show you a visual of actually what a DNA typing profile looks like. Now this is just a partial one that I have up here. This is 4 different sites of DNA that are being demonstrated here. This is a glass stain sample, so a crime scene evidence sample, and it's gone through that whole process. We have done the detection, and now I'm doing the data analysis by my computer software program. This first site of DNA, that is our sex determination site that we look at. And we can determine if the source of the stain was from a male, if it's XY, or if the profile shows up there as XX, then we know we're working with a female that donated a particular stain. And then going on across, we have 3 other sites of DNA on here that have been typed. And the next one here is 1314. That means a 13 came from one of your parents and a 14 came from the other parent. And so it types out as a 1314 individual. And then the reading on across to the next site of DNA that pulls up here, that is what we call a homozygote, as opposed to the 1314, which is a heterozygote. The 30, you only see 1 peak there. Well, that just means that both of your parents contributed the very same DNA, so you are a 30 at that particular peak. And then reading across the last DNA site that we have for comparison purposes is a 1214. So we would take the profile that we generate from our evidence sample, and then we would compare that to our known reference sample. This particular known reference sample is labeled as having come from a suspect. Typically, those are a oral swab sample. They'll take just a swab and rub on the inside of the cheek of the suspect and submit that into the crime laboratory as an actual piece of evidence, and it's called a known reference sample from whatever the individual's name is. We develop a DNA typing profile from the known reference sample, and we can do a direct comparison of the typing profile. And as you can see, these two are a perfect match. So we have a matching pattern between the glass stain sample and then the suspect sample, because every single one of the sites of DNA do match up. Now for comparison, here's another one. It's the same glass stain sample, and they had another suspect on this case. It's also coming from a male, as you can see by this first site of DNA. But even just going to this very next site of DNA, the person who's the second suspect is an 1113, and that does not match up with the 1314. And then reading on across, you do have a number of different variances. So the glass stain sample conclusion for this is going to be that it does not match the suspect sample. Now, we don't just type 4 sites of DNA. There are 13 core, what we call core CODIS loci sites that we look at in the crime laboratory. And we also are looking at that amylogenin site, so a total of 14. And it's by looking at this whole total picture that we're able to come up with statistics that are very powerful. Because as I showed you on that previous slide, if you're just looking at 1 or 2 sites of DNA, you might by happenstance just happen to have the same DNA typing profile between both suspects. But once you've looked at a variety of different sites and the variability that is within each particular DNA site, usually we're able to provide very powerful statistics. Now, by that I mean usually it's basically the terminology is the chances of an unrelated individual person chosen at random in the general population having a matching profile to what we've developed. It's usually something in the quadrillions or quintillions. So that's 170,000 times the Earth's population if it's 1 quadrillion, and many times we do bump up into the quintillions based on our typing profiles. So if we're coming up with a DNA matching profile and it's a 13 DNA site match, you better have some explanation of why your DNA is there, except for my identical twin did it. Now we have heard that on occasion. Okay, I want to talk also a little bit tonight about the FBI's CODIS DNA databasing program. About a year ago, I left doing DNA casework, which I have really enjoyed over the years, but I also wanted to branch out and try a new challenge, so I became the DNA CODIS manager for the state of Washington. This CODIS database houses forensic unknown DNA profiles from our casework samples, and it also has samples in there from convicted offenders. So we can use this to link up serial crimes by matching evidence crime scene samples to each other, and then we can also help to provide information to investigators on, if we have an evidence sample, we run it against our database and it matches a known convicted offender in our database, then we can provide that information also to the detectives. It's a pilot project that began back in 1990, and I'm proud to say that Washington State was one of the 14 states in the country that was asked to initially participate. And now the DNA data is linked in all 50 states across the United States. Washington is now what we call an all-felon state, which means that anyone convicted of a felony in the state of Washington must supply a DNA sample to the crime laboratory, and it's an oral swab sample, like I talked about the reference samples previously, for typing purposes, and that will be entered into the DNA CODIS data bank. Our law also covers 3 gross misdemeanors, which are stalking, harassment, and communicating with a minor for immoral purposes. We get lots of samples. We get about 2,000 samples in a month. So these are initially processed by logging in the pertinent information to our laboratory information management system, and then they're prepared for doing the DNA analysis on that. Once the DNA sample has been typed, it will be entered into the CODIS database. Currently, Washington State's database, we have about 85,000 convicted offender DNA profiles in there and about 1,200 crime scene samples that have been developed. At the national level of CODIS, which links, as I said, all 50 states, it also has Puerto Rico, the FBI, and the Armed Forces DNA Identification Laboratory. There are nearly 3 million convicted offender DNA samples and about 130,000 crime scene samples. An interesting tidbit too is that CODIS is branching out, and it's being installed in a number of different countries worldwide. So not only can we do searches at the state level with our own database, and at the national level with the national level of CODIS, we're able to sometimes, if need be, we can request the help of Interpol and do searches in other countries. And we've done searches over in Canada, England. London has a very large database, and there's about 42 different countries that do participate in Interpol searches. So that's another complement to this powerful tool that we're just adding on. These searches are conducted routinely. The casework analysts can conduct them on their own DNA profiles that they develop off of crime scene evidence at their will. I've given them the power to be able to access the CODIS system and search their own DNA profiles from the crime scene evidence. And if it does match with a convicted offender profile, we will pull that convicted offender profile sample, and also we will retype it again to ensure that there are no possible ways that any mistake could have been made. And then once the confirmation step is done, we will release the name of the convicted offender and the date of birth information to the detectives on the case for them to pursue that lead. Besides typing, putting in the typing results and coming up possibly with a match with a convicted offender, the other type of match that we could have would be 2 crime scene samples matching, or sometimes even more than 2 crime scenes. If you have, for example, a serial rapist being active in your area, and it could be actually active like up and down the West Coast along the I-5 corridor or something. This is a way for our states to communicate with each other through the use of CODIS. We've had about 350 hits in our CODIS database since we came online with this in 2001. And by coming online, meaning that the whole national program is all up and going. So we're very proud of that, and we're working hard to get even more hits to keep everybody safe out there. To end up the evening tonight, I just wanted to talk a little bit about one of the cases that I worked on before I went over into the CODIS section. This is a homicide case. The Seattle Police Department has a cold case squad that was developed in recent years for detectives to go back and look at some of the old cases. Usually they're homicide cases, and they're usually anywhere from within the last decade to 20 to 30 years old. We've actually had one in the crime lab we had success on that was about 30 years old. So this one was, a victim was strangled and raped. The young lady's name was Angelita Axelson. She was found in a Seattle hotel room on June 18th of 1981. Semen was identified by the medical examiner, and that was documented very carefully in the ME report. But there was no suspect list. They just kind of was a dead end. They did what they could at the time back then for investigation, but they really never were able to solve this case. So a couple of the detectives from the Seattle Police Department Cold Case Squad were reviewing case files and determined that this one, hey, they identified some intact spermatozoa by the ME report. Maybe this is something the crime lab can help with. So they submitted it to us for work on that. Darn, this is what I got into the crime laboratory though, and as you can see, these are really kind of nasty brown looking swabs here. I did give it the old college try and tried the extraction, etc., but I was not able to determine a DNA typing profile off of this because the swabs had degraded over the last couple of decades. But since I had read through the medical examiner's report, I was thinking, gee, I wonder if that's all there is on this particular case. And that's often a question that we will ask the detectives because we do work very closely with them. And fortunately, by reading that report and knowing that they had found the intact spermatozoa, I asked, well, is there any chance that they still have the slide that they had looked at back in '81 when they were doing their autopsy? And sure enough, they actually still did have it on the shelves up there. So they submitted it to the crime laboratory. It's sort of a blackish, purplish color here because it's been stained to be more easily visualized under the microscope for the cellular material. Now, it was received into the crime laboratory, and basically I just popped off the top of there's a cover slip covering the cellular material there, and just stick it in the freezer for about 20 minutes and then run a little knife blade under the end and the cover slip pops off, and then you have exposure of the cellular material that's on the slide. So I took that cellular material, scraped it off, and put it into a little extraction tube, made my smear, and saw spermatozoa just like they had seen 20 years ago. So I thought, well, okay, maybe this has been preserved a little bit better. It was in a nice dry state because it was a smear that had been prepared and just left, I believe it was at room temperature all those 20 years. So after I was able to complete those 5 steps of the DNA typing process I talked about earlier, I was able to get nearly a full STR DNA typing profile off of this vaginal smear sample. Now, once again, as I mentioned earlier, this was a cold case. They had no idea, who possibly could have committed this crime. So they were sort of at a dead end. So I did take this DNA typing profile I generated from the vaginal smear and searched it in CODIS and got some pretty exciting news back, which was a cold hit, which means it matched up to one of our convicted offenders that was already in the database. This convicted offender was in our database because he was the bus stop rapist. He was convicted of a series of rapes up on the north end, and when he was required to go to prison, he had to submit a DNA sample for us, and we were able to get a profile from him. And then as I did the search on the evidence sample, they matched perfectly. So when the detectives went over there to talk to the individual who was currently incarcerated at the time, the detectives were able to say, hey, do you know this young woman? And showed them a photo montage of some people, and he's like, no, I don't know any of these people, and kind of digging himself a hole basically, because then he has no way to explain how his spermatozoa got into the victim's body. And this is the gentleman that did end up pleading guilty, Brian Jelena, to the murder of Angelita Axelson. And it's these kind of cases to me that are really satisfying to work on. I mean, not only is it a challenging type of evidence, but it's a crime that's many decades old and kind of forgotten about with a lot of people. But the loved ones of that particular person have never forgotten. And the other important tool or thing to remember about these kinds of cases is oftentimes there'll be sort of a cloud of suspicion that will follow, either a neighbor or a boyfriend. Detectives and or family members might think so-and-so did it. And fortunately, by being able to help solve the crime through the technology of DNA evidence, not only can we help to show who actually is guilty of the crime, but we can help to lift a cloud of suspicion over somebody's face that is really shouldn't have had it over themselves to start with. I think my last slide is here. Appreciate your time tonight. These are some different websites that you can actually go to. There's a whole plethora of information out there on the internet right now. This is just a couple of them out there. If you Google anything about forensics or CODIS, you'll just find tons of information. So thank you very much for your time.
I'd like our presenters to come up, and then please use the microphone, and let's open it up to questions.
I have a question about the CODIS system. Has there been any research regarding the felons in the 13 areas that you look at? Is there any similarities there compared to maybe the victims in the case?
Well, the 13 sites of DNA we look at, those are the same 13 sites we would look at whether you're a victim or a suspect, unidentified human remains, whatever. So there's no correlation. It's all just inherited genetically from your parents. We have, you don't start out to be like a bad person or a good person or anything.
Okay, thank you.
Was there anything that you've learned through what you've done that was so unequivocal that it's changed the way that you live and the things that you do, or you would at least recommend to this group that they should change?
Well, look, I can answer that to some extent, I guess. One of the things I was talking to somebody at the break about was because of the exposure that I have and what I see in terms of traffic accidents and fatalities, I just drive extremely defensively. I'm always thinking that person coming towards me, or if I come to an intersection, that other person who's coming to the intersection, I don't know anything about them. And you always have to anticipate what somebody else might be up to. So I drive very defensively, and I'd encourage anybody else to do the same.
There are so many things that you could think about. I think two things. One is that one of the things about forensic pathology is you actually never know what's going to happen to you tomorrow or tonight. So you really have to live your life and in a good way. And so I think that's an important thing. Most of the people who die suddenly and unexpectedly or due to violence didn't expect for that to happen. So that is some way that you can think about as a guiding light for your life. I would say as a personal thing, I actually think you should always be careful when you cross the street. And you should never cross in front of a car that doesn't stop before you step in front of it. But, and we laugh, but there are a lot of pedestrian injuries, and those drivers are running into people like us who are walking too, so.
Yeah, I would just add, having moved to Seattle 3 years ago, I was struck by people not realizing the physics of who wins when human interacts with car. Yeah. And people just step in front of cars with an expectation they'll stop.
Right. You can be right or you can be dead right.
So I think that is an important issue. Other questions or comments?
I would expect that a bunch of us, if we haven't already been called for jury duty, will be called at some time. And my question is for any or all of you. What is it that you would like jurors to either realize or understand that they don't seem to?
A great question.
I can speak to a couple of things there from having testified over the years. Sometimes I think there's an unrealistic expectation of what can be done by science. We maybe have the technology to do all this fancy DNA typing and trace analysis and et cetera, but there is a limited amount of resources and time involved. So we basically get in there and try to figure out what are the key questions that need to be answered. And sometimes you can do that by just looking at one piece of evidence. And sometimes you may need to look at 100 pieces of evidence. And actually, sometimes the crime lab doesn't need to be involved at all. I've had people come up to me and say, well, I was a juror on a case recently, and the crime lab wasn't even there. We were waiting for some evidence from you guys to be presented. But really think about what the case is, and it's the prosecutor and defense attorney's job to put it all together. I guess that would be one of the key things that I would hope a juror would understand, that what you see like on TV, that everything is tested or whatever, is not realistic.
Thank you. Please.
I was just interested about the constraints that you have about regarding the use of the DNA. Is it very expensive to have these tests? Or you mentioned that you don't have much time. Is it a matter of personnel or time or expense or just what? I have the feeling that it isn't used very often, but just in very few cases. And I just wonder why it isn't used more often.
Actually, it is used in a wide variety of cases. We do have a tremendous backlog across the state of Washington, and it's not just the state of Washington, it is nationwide. There's national funding that is being supplied, but the dollars is not going to be a panacea for this huge influx of these types of cases. The exciting part is I'm able to tell you all these different types of body fluids and even the cellular type of material that we can get DNA profiles off of. But the downside to that is detectives are realizing, and prosecutors and defense attorneys, how valuable DNA methodology is. And so they keep, shoving more and more items of evidence into us, and we're doing the best we can to keep up, but it does take about a year to a year and a half to train a new DNA scientist coming out of a university. So it's a lengthy process.
I believe this sort of follows with what she just said. There was something, I believe it was in the Seattle Times, was it today or yesterday, about how the state of Washington seems to be lagging behind some other states in doing this. I mean, Oregon, I guess, just hit their 1,000th hit or something, and here we are at 350. I was wondering if that's a funding issue that you're talking about?
I know in Oregon, when I talked earlier about our forensic crime scene samples being at about 1,200, in our state database, they are sitting at about 3,000 samples in their database. They've put a priority apparently on testing burglary samples. So it's a goal of ours to have a high throughput system so that we can analyze many more burglary samples, but it does take some time, funding, and like I said, most critically is just the personnel, because we can't just hire somebody coming in off the street, even if they have all these degrees and credentials. Forensic sciences is something where you actually learn an awful lot of it on the job, and it takes a long time to do a quality piece of work. So there's also I would have some question with them as far as how they actually are counting their hits are not exactly the same as what I'm reporting here. So you have a little bit of discrepancy on the numbers game, and that actually goes on nationwide too.
I can add something as far as our resources in the state of Washington are concerned, is that we've been working with the legislature for the last 4 years to improve our facilities and resources and our staffing. We've built in the last 5 years, we've built a new laboratory in Seattle, new laboratory in Vancouver, Washington, and a new laboratory in Cheney. And we're in the process of training new staff in those laboratories. And the budget that the legislature passed today included $3 million in funding to increase the size of the DNA laboratory in Vancouver. So the state is investing in these resources. As Jean said, it does take a while to turn things around, because it does take about 9 months to 12 months to train somebody before they can start doing the kind of casework that she was describing. But we are certainly making progress.
I guess with that, I'd like to say that over the last 2 years, we reviewed what you all have asked to see, and forensic medicine and pathology was something folks stressed. And I had an amazing teacher of pathology in medical school, and it's actually the first time that you get to see that if you pay attention, you can put things together and figure out disease. And we thought it was a nice juxtaposition of last week's talk, where even after someone dies, an organ transplant or education, you can have a big impact on the living. And I think we demonstrated that nicely again today in education and affecting how we think about causes of death. So thank you all very much, and thank you.